eventually it is sent to the central nervous system for processing. What happens at the transduction site? This is the assumption of what happens. So when imagine this is a peripheral nerve terminal. When there is a stimulation at the tip of the terminal, current will be activated by a channel opening, mechanosensitive channel opening. And then with the current flowing in, there will be a depolarization of the membrane. And when the membrane potential is exceeding a certain threshold, action potential is fired. What is largely unknown is how the mechanical response is generated, how the current is generated. One of the well-characterized models in C. elegans. In this C. elegans, it is found that the transmembrane mechanosensitive channels, MEC4, MEC10, they are tethered intracellularly through a link, MEC2, to the cytoskeleton, MEC7, MEC12. And extracellularly, on the other side, tethered by MEC1, MEC9 to the extracellular matrix, MEC5. However, in mammals, it still remains a big question mark. So far, only our group has identified a MEC2 homologous, which is SLP3. It is required for the mechanotransduction. But the other part, like the tether and the ECM and its molecular nature, is poorly understood. So the aim of my project is to study the molecular mechanism of touch in mammals, and also what happens at the cellular level. It's extremely difficult to study this because the transduction site is at the surface of the skin, and the soma, the cell body of dorsal root ganglion sensory neuron, is here in the back. So in order to do that, we have to we also established an in vitro physiological assay for mechanotransduction. What we did is we cultivate sensory neurons derived from the DRG and played it on a cover slip pre-coated with EHS laminin. EHS laminin is it contains a mixture of different laminin proteins, laminin 1, 2, 3, 4, etc. And when there's neurite growth, we will poke the neurite, stimulate the neurite, and then record the current from the soma. And basically, 3 types of current are recorded based on their inactivation time constant. We classify them into rapidly adapting, which account for about 40 to 45% of all neurons, and intermediately adapting, about 5 to 10%, slowly adapting, about 40 to 45%. There are also